Myself, Mr. Mate H.T., Department of Botany, RBNB College, Sri Rampur. So today we are going to study the T.Y.B.Sc Botany, that is semester 2nd, paper 1st, BO 121. And the name of the paper is Plant Life and Utilization. And from this topic number 2, that is the Pteridophytes. And from this type, there is one type or specimen which is called as the Nephrolipis. So we are going to study the life cycle of the Nephrolipis. Now from this first, that is the habitat, that means where these Nephrolipis species are found. All over the world there are about 30 different types of the species of the Nephrolipis are found. Now the habitat, it is evergreen, it is a perennial, it is a terrestrial, and it is occur in humid forest. That means where humidity is more. In such a condition, the species of this Nephrolipis are get developed. So that is about the habitat. Now habit, that is the external morphology of the Nephrolipis. Now it shows the different characters. Now first character, the plant body is a sporophyte. That means this plant body of a Nephrolipis is called as a sporophyte because this gives the spores. These spores are the reproductive structures. Now these spores, they are developed in the sporangia, and these spores get developed into a next generation which is called as the gametophyte, which gives the male gamete and female gamete. That means a sporophytic generation which gives the spore is called as the sporophyte. It is a perennial. It is a dominant, that means it is a dominant in its life cycle, because there are the two different generations, that is the sporophytic generation and the gametophytic generation. So this is the sporophytic generation and it is dominant in the life cycle. And this Nephrolipis is get differentiated into the different part, just like the higher plants. For example, it is get differentiated into root, stem and leaves. So this is the roots, this is the stem, and these are the leaves. Now next character, the root is adventitious. They are developed on stolon. Roots are very small, they are fibrous, they are branched, and they also show the root hairs. So these are the adventitious roots. They are very small and they have the root hairs. Now next, spur develops the new plant. Now in this plant Nephrolipis, there are the spur. They are important for the natural propagation. So this is the spur from which the new plant is getting developed, because from upper side the shoot system is getting developed and from lower side the root system is getting developed. So these spur, they are important for the natural propagation or the vegetative propagation. Now stem is erect, it is short, it is thick, it is condensed, it is cylindrical, and it is known as the codex. So this is the stem. It is a very small, condensed, and it is oblique, and this stem is also called as the codex, and it is underground. And on this there are very small structures are developed. They are called as the scales. Now stem gives the large number of the extra axillary branches, and they are called as the stolon. Now these are the stolon. Now they are also important for the natural propagation because they develop the spores. They develop the spurs. Now these stolons are called as the extra axillary branches because they are developed from the axil of the scales, and so they are called as the axillary branches. They are known as the stolon. Now next, each leaf is differentiated into basal part, which is called as the stipe, and upper part where the leaflets or the pinnae are developed is called as the rachis. So this part of the leaf is called as the stipe, where there is no development of the pinnae or the leaflets. But the upper part of this stipe, where the leaflets or pinnae are get developed, and they are called as the frond, or it is also called as the rachis. So this is the rachis, this rachis, it is also called as the frond. That means when the leaflets are developed, then that rachis is also called as the frond. The rachis bears the leaflet or pinnae from both the sides. When a leaf is young, it is inverted to one side, and it is called as the circinate leaf. So this is the leaf. It is in young condition and it is rolled to one side only. Such type of leaves are called as the circinate leaves. Leaflets are sessile. They have auricle, lanceolate, and have acute apex. So this is the complete leaflet, and it has a auricle. It is a lanceolate, and it has an acute apex. It is lanceolate, that means it has a shape is a lance-like, so it is called as the lanceolate. And the apex is acute. And they are sessile, that means that they do not have the stalk. The leaflet or the spinni are directly get attached to the rachis, or they are developed on the rachis, they have no stalk. Each leaflet has a distinct midrib and it is a forked. Now here, this is the mid vein, and this mid vein shows the lateral veins, and these lateral veins are forked. That means they show the bifurcation. So this is the bifurcation in the lateral vein which are developed on the mid vein. And at the end of this forked vein, there are a special type of structures, and these structures are called as the Hydathon. So these are the Hydathon. They are important for the process of guttation. So this is the structure of the Hydathon. Now this is the vein ending. That means where xylem and phloem are developed. Now this xylem gives very large amount of the water in this next part, which is called as the epithelial cell. So at the center there are very large number of the epithelial cells. They are thin walled and they are loosely arranged. Now water from this vein, that means from xylem, enters in this epithelial cell. They are hygroscopic in nature. And from this hygroscopic epithelial cell, water is given out through the water pore or through the water stomata in the atmosphere. And toward the periphery, there are the normal epidermal cell. So all of this is the stem, this is the stolon, this is the spur, then these are the adventitious root, and above this there are the leaves. Leaves are compound and they are pinnately compound. Now this is complete external morphology of the Nephrolipis plant. So the next point, that is the internal morphology of the Nephrolipis. Now Nephrolipis shows the different types of the parts. So from this, the number one, that is the internal structure of the stolon. So this is the complete internal structure, one patch of the internal morphology of the stolon. Now it is somewhat circular in outline. Externally it is covered by a thin layer of the cuticle. Now inside this there is a single layered epidermis. Cells are parenchymatous, and this outermost layer, that is the epidermis, it is a protective in function. Inside this there is a multi-layered cortex, but this cortex is get divided into the two parts. The upper part or exterior part is called as the hypodermis. So this is the hypodermis. The cells are lignified, they are compactly arranged and they are thick walled. So they give the mechanical support to the stolon. So this is the outermost part of the cortex. It is a 3 to 4 layers and all the cells are thick, they are compactly arranged. Inside this there is the layer, another layer of the cortex. Now most part of the stolon is occupied by the cells of the cortex. They are thin walled, they are loosely arranged, and they are parenchymatous in nature. They are somewhat circular in outline, and as they are parenchymatous in nature, they store the food material. Now the innermost layer of this cortex is called as the endodermis. So this is the endodermis and it is a single layer. Now inside this there is the still. Inside the endodermis there is the still, and just inside to the endodermis or toward the outer side of the still there is a pericycle. It is a 2 to 3 layered. So this is the pericycle, it is a 2 to 3 layered. Now inside this, at the center there is the xylem. Now xylem is again shows the two different parts. That is the cells which are small of the xylem, they are called as the protoxylem, and it is toward the periphery. And cells of the xylem which are large, they are at the center, and it is called as the metaxylem. Now this complete xylem is covered by a phloem. So such a type of still is called as the protostele. So the type of still in stolon is the protostele. So this is the complete internal structure of the stolon. Now it shows the vascular tissue, that is this is the xylem and this is the phloem.